Okay, thanks Matt. I'm going to be talking about plankton observations from last year, and it's actually going to be a double act. I'm going to start off and then hand over to Jeanette, and then I'll do the last bit. But before I start, there's a lot of people to thank in this presentation. I particularly want to single out the last line there, because a lot of people have shared observations and photos of particularly gelatinous plankton from around the West Country, and this talk is actually going to be quite a lot concentrating on those observations. So as I said, there's three parts of it. There's quite a lot to get through, so I'm going to be focusing on the single most remarkable observation from the plankton from last year, which was the huge increase in the numbers of gelatinous filter feeding plankton known as salps. They were really incredibly abundant in the last half of last year. So I'm going to focus in a little bit more on salps, then hand over to Jeanette, who's going to be describing the Southwest Jellyfish Survey, and then I'm going to do the last bit with something that Bob asked us to present, which was actually how each person's presentation links up to the other themes in the conference, and a bit of a forward look. And I'm going to be picking up on some of the new and novel plankton technologies which particularly make the link between plankton and fish. So I'll just start off with the plankton, and I mentioned the salp increase, and I just put this slide up to show what salps look like. It's a sort of like a noddy identification guide that I put into the last year's annual report. So the salps have actually got quite a complicated and interesting life cycle. So there's a pointy one, a barrel-shaped one. They go in both solitary and chain forming. But the thing about salps that is quite noticeable if you're kayaking or swimming and you don't get a good view of them, they've got these sort of opaque blobs in the middle of them. That's actually the stomach. They're filter feeders, and they pump water through an internal fine mesh filter, and they can filter down to the size of about bacteria. So they've been described as sort of like vacuum cleaners, which hoover up everything out of the water. So really efficient, and they can form these massive blooms. So they're not to be confused with other chaining gelatinous zooplankton, for example, the siphonophores. So just looking at the observations last year, you can see from this plot the absolute extent of the increase in the numbers of salps in the West Country in the last couple of years. So the blue lines and the y-axis on the left is the numerical abundance that we've recorded in the L4 site that Tim mentioned in the last talk. So you can see a huge spike up to about 50 just in the last year, July, August, but these observations from the Southwest Marine Ecosystems report that Paul compiled, I've scoured the previous reports, they go back to about 2018 where we saw salps, nothing before. So this is a really recent and quite dramatic increase in salps. So the obvious question, why have they increased so suddenly? And a really obvious explanation could be something like the heat waves that Tim just mentioned. Possibly they're like a warm water species, and these the warming climate has sort of brought the salps into the area. That doesn't really hold, because Andrea identified them as a sort of common or garden salp, Salpa fusiformis, and that's actually got quite a widespread distribution. It's in cold and warm water. So that I think we need to look a little bit more widely at this sort of simple temperature type of explanation. So the next thing we looked at was maybe feeding conditions have improved for them. So to look at that, we classified all of the sort of dominant zooplankton taxa that we get at L4 over the last 30 years, and we classified them into three types. Basically the fine filter feeders, for example salps, which strain down to the size of sort of like bacteria, a couple of microns size. Also the carnivores and omnivores. And we looked at the trends over time and ranked them according to the amount they'd increased or decreased on average over the last 3 decades. And you can see there's some sort of shuffling of the fine filter feeders towards the top of the league table. So it's not just the salps that are increasing, but other fine filter feeders, for example, the related doliolids, the very common appendicularians. So something is changing in the habitat that is favouring these fine filter feeders. So this sort of goes along with something that we've been looking at over a couple of years, not just at L4 in the West Country, but more widely over the Northwest European Shelf and the Northeast Atlantic. We seem to be moving away from this food web over time with climate change. We're going from this sort of classic food web, from blooms of diatoms or dinoflagellates that are fed on by copepods, in turn supporting fish. We're going towards this sort of slightly more complicated food web, and we think it's due to warming, which is increasing the stratification, cutting off the nutrient supply, favouring smaller phytoplankton cells, these picoplankton, which are much more efficiently fed on by these filter feeders, for example, the salps and some of the meroplankton. So that's the idea. Personally, I don't think it's the whole explanation for such a rapid increase in salps. I think there's something else going on, but it's definitely work in progress. So I'll stop there, hand over to Jeanette. So there you go, I'll sit down.
Thank you. I'm afraid these slides are a little bit busy, so I apologise for that. So I'm going to talk about sightings that have been reported to us. Mostly these are social media sightings, where we require a photograph and a place and a time, and we try to get an estimate of numbers. Because it's really very variable what people report, we've tried to group them according to abundance. So what you've got across the top are the most common tree jellyfish, and then going down we've got the time series. So the top row is 2023 going down to 2019. And also split it a little bit between the different coasts of the 2 counties. If it's grey, it means that they were seen, but perhaps only 1 was reported, or only up to about 5. If it's purple, it means there was at least 1 sighting where quite a few were reported, so it could have been anywhere between 6 and 50. And if it's black, it means that a large number were reported, but that could be 50, or it could be an entire bloom that was maybe 1,000. So I think the barrel's quite an interesting pattern. What's not on here is 2018, because we don't have enough data for all the different species. But they were very abundant in 2018 and 2019. But the difference between that and 2023 is that our 2023 sightings are all strandings. In 2018 and 2019, they were reported all around the coast, but there were just as many being reported live and swimming inshore as there were being stranded. The other 3 are showing fairly similar patterns, I think, across all the years. It's hard to go back further than that because there isn't that much data. So what I looked at is Russell's summary, which he published in 1970. And these patterns are very much in line with what he would have predicted these species to occur and disappear. What's slightly different is most of these species he recorded as being sort of sporadic, so you might get them 1 year and not the next, whereas we're seeing fairly consistently that they're around most years. The other thing, I don't know if this is genuine or not, but his reports of the first appearance of these species were very much reliant on trawls, and he was talking about very small individuals who are maybe 10 millimetres in diameter. And so what we've got are the same species being reported at the same time, but by people who've taken a photograph or seen them stranded on the beach, so probably larger individuals. So the next slide I've got are the ctenophores. So a lot of the reportings are just saying ctenophores, and you get a blurry picture, and you can sort of see a rainbow effect. You know it was a ctenophore, but you don't know the species. So I've separated out the sea gooseberries and the barodes where they actually were clear from the photographs which species they were. And then on the right, you've got everything combined, including those species. The only thing that I can definitely say is that in 2019 and 2020, the only reportings we had were of strandings, whereas recently, the last 3 years, there have been lots of reports of them in the water swimming. So moving on to the hydrozoa, I think they're quite interesting. So if I talk about the Portuguese man-of-war and the by-the-wind sailor first, they're not really inshore species, so we tend to see them when there have been storms and mostly when they're reported washing up on the beaches. And so they seem to be occurring a bit more in the summer months in this data set, but I don't know how unusual that is. But the crystals had a really interesting pattern this year, a little bit like the salps, and it's not really shown by this data. You can see that there were several reported in August and September, but I've just put up some maps to kind of show this distribution. Unfortunately, with the maps, my colour scheme didn't work, so I had to change it a little bit so that white means that there was just a few reported. Then again, purple is still that 60 to 50, and red is over 50. And you can see that by August, there was a large amount being reported in the Channel Islands. We did have quite a lot in Newquay, but by September, again, they were being reported all along the south coast and up round to Lundy in large numbers. So, moving on to the salps, I think I just really like this photo. It really shows you what Angus has been talking about with the salps. So this was taken in Cornwall, so there were a large number reported and consistently reported, so not just on one day. You can see from our records on the left-hand side, they have been reported before, but they were very localised. So if we look at some of the maps, you can see here that they were being seen in August from Plymouth all the way round to Hale. But by September, they were still being seen in large numbers all the way around the south coast. Okay, rarities. So these photographs are all from 2022. The only one that was seen again in 2023 is the one on the left-hand side. The others weren't spotted again during 2023, but we have just had some siphonophores spotted off Cornwall this year, so maybe this year. And I'm going to hand back to Angus there.
I have a sneaking suspicion we're going to run into time trouble, so I'd better get going. Right, this is the last bit of the talk, and it's actually making the link to some of the other topics that we're going to talk about, particularly the fish. I think there's 2 fish talks later today, but it's looking at novel plankton technologies. And the traditional way we look at plankton is to use bottles or nets and then take them ashore and analyse them through a microscope. And that's formed the backbone of all the really good long time series observations of plankton that we've got. So it's basically forming our understanding of how they have responded to climate change. But with new technology, there's a lot of different ways of looking at plankton, and a key one that's emerging in the last few years is imaging. There's a whole variety of approaches to plankton imaging, and I've given some of the advantages and the drawbacks of the different methods in the blue and the red text. Haven't got time to go into it, other than to say imaging is really good, but we haven't yet built up the long time series from the standard method from imaging to understand climate change responses. Likewise, molecular, brilliant technique. It reveals the so-called hidden diversity and some of the rarities in plankton, for example, eDNA and metagenetics. Likewise, it has got short time series. It's a rapidly developing method. But the thing I want to show in this slide is that it's the intersection of these methods. When they're used together, that's when you get the best insights into the plankton. And I should say at this point, what Jeanette has just described, really good for upscaling, getting the regional coverage that is not provided by these fixed point time series, and it's also looking at some of the larger taxa. So the key take-home of this message is that there's a lot of sort of buzzwords in here like AI and eDNA, and a lot of policymakers and funders have actually latched onto that as if these novel techniques are a sort of cheap solution, so sort of like abandon expensive time series and continue with eDNA or artificial intelligence and imaging. The point is with this, they should be used actually together and then they complement, because we definitely need to combine the different methods. Equally important, we need to retain the taxonomists who are absolutely underpinning all three or all four of those methods, and we need to continue the consistent identification of time series. So at L4 we'll right, I've really had better speed up. We've got imaging techniques, and this has been going for about 10 years or so with the flow cam. That's really good for picking up the intermediate size organisms between what is sampled with bottles and nets, and it also makes beautiful images which are classifiable for some of the more delicate and gelatinous taxa that are missed by the two methods. And these are being developed at the moment at L4 with a system called APIX, which is actually a dual imager. The instrument on the left is measuring the small size fraction, the one on the right is measuring the large, and that's giving us a really good high frequency picture of the dynamics, for example response to storm events or tidal mixing, of the complete size spectrum. There's also molecular approaches. They've been running for about 11 years now at the L4 site, and I haven't got time to go into the details of it, but there's both 12S sequencing for fish and 18S for the zooplankton. I think Karen Tate and Penny are in the audience, so I'm not a molecular person. Either Karen or Tate can address any technical questions about it, but I just want to follow on the use of the eDNA in these surface water samples that are filtered to detect fish. So they're detecting quite a much higher diversity of fish than concurrent trawl samples, but I think the most interesting thing is even over 11 years they're detecting significant trends in the molecular diversity of the fish component. And there's some winners, for example bass and the pilchard, but there's a bigger list of taxa that are declining in their molecular diversity at the L4 site. And just to go into this in a little bit more detail, this is, I think, an issue with the molecular is actually transferring the genetic information to abundance, and this is using a proxy of abundance called amplicon sequence variance, and that's basically intraspecific molecular diversity, and this is increasing, for example, for the sardines and the bass, and the heat maps on the right are the seasonal picture as well as the long term, but for a wider list of species it's declining. So this is a really obvious link between the plankton samples and understanding changes in fish. So I'll just quickly go on to the conclusions. High SELP numbers, we've talked about that. Linkages, making the link between changes in plankton and the supportable biomass of fish, but also using these surface water samples, the so-called eDNA approaches, to get insights into trends in fish, which are really, really tricky to get with other methods. So we're not saying the molecular is better, but in comparison with landings and other census data, hopefully all the pieces in the jigsaw can be put together to understand the trends in fish. So I'll stop there. Thank you.
Thank you, Angus and Jeanette. And I couldn't agree more.